In the menu, navigate to Molecular Biology Tools, to Gibson Assembly. In my basic setup, you can see the scenarios indicated as single set assembly. However, you can also do combinatorial assembly and split sequence. Single set assembly is when we can design DNA fragments with complementary overlapping ends, allowing them to be joined seamlessly when synthesized. You can also see that there are other fields you can adjust based on your specifications. Click Next. For my backbone linearization method, I will keep PCR. However, you can also select Restriction Digest. For initial overlap mode and terminal overlap mode, there are different selections as well. I will upload my backbone. And the fragments. To the right, I will select the GFP. I will do the same with the fragments. Now you can see each assembly part has a fragment start and a fragment end, as well as junction sites 5' and 3' for the backbone. Run design. Here you can see my design schema with the 3 assembly parts as well as the backbone. The primers here, homology overlaps, primers in the raw text format, and complete amplified fragment sequences. Click Assemble. To the right, there is the assembled construct here in the graphical map. And as I scroll to the right, you can see the assembly parts inserted into the plasmid. You can export to CSV, send to plate, or register all. Register all will register both the construct and the primers. You can select this. Now I will run an advanced search. I will search based on date created and run search. Here is my DNA part. To the left, you can see the registered construct. To the right, you can see the sequence. The annotations. Here are the bioregistry sequence annotation, the label, start position and end position, and annotation type. Seamless Gibson assembly with optimized single-set reactions in an electronic notebook like this one facilitates precise fragment joining and accelerates complex DNA construct development.